let's start with the ionocyte discovery. I'm going to introduce a little bit about the background. So this is a study led by Harvard Medical School. The study is trying to create a single cell map of the airway epithelial cells to let us to investigate gene expression across epithelial cell types. And this study made a very surprising discovery of pulmonary ionocyte. And the pulmonary ionocyte showed very unique gene expression profile and really like highly enriched in ion transporters. Some of the key features of pulmonary ionocytes are including one of the key transcription factor, FOXI1 gene, being required for ionocyte specification, and also there's a key finding is the cystic fibrosis gene CFTR is highly enriched in ionocyte. And there are also other ion transporters like ATP6 V family proton transporters in pulmonary ionocyte. And the gene expression profile in those pulmonary ionocytes showed a very high similarities to fish gill ionocyte, frog skin ionocyte, and kidney intercalated cells. Well, the ionocyte in those species and organ has been well studied. So their function are majorly in ionic homeostasis regulation and osmotic regulation. So this made us to think about ionocyte play a role in fluid regulation on the surface airway epithelium. So then this led into one of the central hypotheses on pulmonary ionocyte. So we hypothesized that pulmonary ionocyte play a key role in maintaining the airway ion and the fluid homeostasis through CFTR. Well, to study cystic fibrosis and CFTR, it really involves both human patients and animal models. And we are very lucky to have different animal models in the field to study CF, like ferret, pig, and sheep. So talking about the airway cellular anatomy, they are quite different across the species. For example, the human airway contains 4 trophic units, including the proximal cartilaginous airways and the terminal bronchioles, respiratory bronchioles, and some mucosal glands. While the ferret pig, they have very similar trophic units as compared to human. And other species such as mouse and rabbit, so they are very divergent. For example, mouse, they just have some mucosal glands very restricted to the proximal end, and rabbit does not have some mucosal glands at all. So a pulmonary ionocyte, they present in proximal cartilaginous airways and also in some mucosal gland. So our group, we use ferret as a model to study cystic fibrosis because ferrets, they spontaneously develop CF lung disease phenotype. So now I'm going to talk about some of the study we did regarding to use CF ferret models to address the ionocyte biology controversy and the ionocyte function stuff. The first study we did is by using single cell RNA sequencing to create a single cell map of the ferret airway epithelium. So from this study, we found all the major cell types, secretory cells, basal cells, cyclin basal cells, and synthetic cells present in the ferret airway epithelium. And we also found the royal epithelial cell types, such as tuft cells, neuroendocrine cells, and pulmonary ionocytes. So this is a data set which allows us to investigate the gene expression across all the ferret airway epithelium cell types. So now we check into the gene expression profile. So a lot of very surprisingly, so the pulmonary ionocyte showed a really high concentration of ion channels. For example, the proton pumps, ATP6, V1G3, and many other proton pumps, ATP6 family, and also chloride transporter, such as bisonatol chloride transporter, CLCNK. And most importantly, the cystic fibrosis gene CFTR is very highly enriched in ionocyte. Well, those ionocytes also showed a very high abundance in other ion channels, such as calcium channels, sodium, potassium channels. So, so the next we just check out a few transporters by using protein staining, because the study I just showed are all at the gene expression level. Then go to the protein staining. We check a few ion transporters. One is the proton transporter, and the CFTR, the chloride transporter, which is associated with cystic fibrosis. So what we found here is that we found a very good colocalization of CFTR to ATP61G3. They perfectly colocalized into the pulmonary ionocyte, and the CFTR showed a apical cap expression pattern in the ferret trachea. The next, we checked other ion transporters. So we did a co-staining of CFTR and the BSND. BSND is the subunit of the CLCNK and CLCNKB, which is majorly involved in salt absorption. So we can clearly see the very good colocalization of CFTR and the BSND in ionocyte, and the CFTR in the apical cap pattern in those BSND positive pulmonary ionocytes. Then the other ion transporters we investigated here is NKA, which is sodium potassium ATPase. So it's an enzyme which help the sodium potassium translocating across the epithelium cell membrane. And NK is a key player in salt secretion in fish gill ionocytes. So then the co-immunostaining of NK and ATP61G3 results show that they just perfectly colocalize into the ionocyte. And we found a very unique structure of ionocyte is that the NK expression are very highly enriched in those apical rims, as we can see in those images. Then next, we just did a comparison of the ferret pulmonary ionocyte to those fish gill ionocytes. So we can clearly see they showed a very conserved ionocyte structure. For example, they showed those NKA tubular extension, and as I mentioned, they showed NK highly enriched in those apical rims when you look at those high magnification images. So the ionocyte structure are like very conserved across species. So pulmonary ionocyte are enriched in ion transporters like ATP61G3, which is a proton pump, and CFTR, NKA, and CLCNK, those transporters. So next, we're more curious about how ionocyte located along the respiratory tract. So we look into the very proximal end of the respiratory system into the nasal cavity. We checked two places here. One is the nasal respiratory epithelium, and the other is the nasal olfactory epithelium. So we use BSND as an ionocyte marker, and we found the ionocytes in nasal epithelium showed very high density, and the structures of ionocyte are quite different when you compile the nasal respiratory epithelium to the nasal olfactory epithelium. In the nasal olfactory epithelium, the ionocyte showed more like neuron structures. Then next, we check the CFTR expression, and so this is a high resolution image to show the CFTR are very concentrated in the apical side of those BSND positive ionocytes. The next, we made a comparison between nasal respiratory epithelium and the tracheal epithelium on the pulmonary ionocyte numbers. So as you can see in the image, the nasal respiratory epithelium showed a much higher ionocyte numbers when compared to trachea. Then we move to quantification. The results demonstrated that in the nasal epithelium, the ionocyte numbers are much significantly higher when you compare to trachea epithelium. So this study really inspired us to generate a cultural system with very stable ionocytes. So here we use ferret nasal basal cells to generate the L liquid interface cultural system. As we can see in the image, this cultural system really shows very high density and stable ionocyte numbers in those wild type ferrets. And when you move to the FOXI1 knockout, so as I mentioned, FOXI1 is a key transcription factor which is required for ionocyte specification. So in the FOXI1 knockout, we can see the ionocyte is completely lost in those cultural system, which is very consistent with FOXI1 being required for ionocyte differentiation. The next way, we check the CFTR protein expression in those cultural system. So we can clearly see the CFTR protein are very highly concentrated in those BSND positive ionocytes. So those really give us a good model and a tool to investigate ionocyte function. So then next we are trying to address how ionocyte contribute to chloride movement across the epithelium. So first we tested whether ionocyte are contribute to chloride absorption. So this is a study that in a human airway culture by using a Wilson chamber assay, and they overexpress the FOXI1 gene, which can increase the ionocyte numbers, and then put a cultural system into a chloride gradient condition, which with apical high chloride and basolateral low chloride. So we can clearly see that the ionocyte overexpressed the human cultural system showed a good amount of ion moving, chloride ion moving from the apical side to the basolateral side. Then just make a comparison of the ferret modeling system to a human model system. We did a very similar studies. We put the cultural system in first into the symmetrical high chloride condition, then move to a chloride gradient with high apical chloride and no basolateral chloride to make a chemical gradient to drive chloride absorption. So what we can find here is that in a wild type cultural system, they showed a very huge amount of chloride ion moving from the chloride to the from the apical side to the basolateral side, which is stimulated by phosphocholine and IBMX and inhibited by glycogen-1. And the FOXI1 knockout group, so it's an ionocyte depletion group, showed the absence of chloride absorption by those Wilson chamber assays under short circuit current conditions. Then go to the quantification, we can clearly see the ionocyte depleted cultures showed a significantly reduced chloride absorption. The level is close to zero when you compare to those wild type cultures. So those graphs are CFTR activation and inhibition conditions on short circuit current change. So then we made a conclusion on that pulmonary ionocyte regulates chloride movement from the apical to basolateral side, which is in an absorptive direction. So the next we are trying to address how do ionocytes regulate fluid absorption. So this is a study our group did 7 years ago. So the method here is pretty simple. So we use the fluorescent dye into the outer surface liquid and use those small volume of the fluorescence outer surface liquid to challenge the human bronchial epithelial cells. Then we measure the chloride, the outer surface liquid height in a time course manner. So what we can hear is that in a human CF epithelium, they showed a much slower fluid absorption when you compare to those non-CF human cultures. And by quantification, the fluid absorption rate is very significantly reduced in the CF cultures as compared to non-CF. Then move to the ferret modeling system, we did ionocyte depleted culture. What we found here is the ionocyte depletion showed a much slower fluid absorption when compared to the wild type cultures. So then we conclude that pulmonary ionocyte regulates fluid absorption. So the next question we're trying to get into is about the molecular mechanism. We ask about how does chloride and the fluid absorption dependent on the ionocyte-specific CFTR gene. So we reported two transgenic ferret lines. One is the FOXI1 CRIA-T2, by which we insert the errors on CRIA-T2 into the 3' UTR of the FOXI1 gene. So this is an ionocyte reporter ferret strain. And the other one is the CFTR conditional knockout. We flux one of the CFTR exons, which is exon 16. So the double transgenic ferret FOXI1 CRIA-T2 CFTR flux, we're able to specifically delete the CFTR gene in pulmonary ionocyte when we expose either cultural system or those animals into tamoxifen. So then the first thing we did here is to check the CFTR deletion in ionocyte in those conditional knockout animals. We have two groups here. One is the vehicle, which is a control, and the other one is the tamoxifen treated. So we can clearly see in the control group, they are CFTR positive and BSND positive ionocytes. And in the conditional knockout group, the CFTR apical cap expression pattern is depleted. Then next we go to a more like quantitative method, Western blot. So we can see here in a Western blot, the control group shows the CFTR C band, and the conditional knockout show the absence of the CFTR C band. Then next we tested how the ionocyte-mediated chloride absorption depends on the CFTR. So this is a very similar study as we did on the FOXI1 knockout of chloride absorption studies. So we have three groups here. One is the control and conditional knockout, which is made by tamoxifen treatment, and we use cystic fibrosis ferret as a CF control here. So we put those trans walls first into the symmetric high chloride, and move into a high apical and low basolateral chloride. So what we can hear is that the control group showed a very large amount of chloride moving from the apical side to the basolateral side. And this chloride ion movement is completely abolished in both conditional knockout and the CF group. Then by quantification, so we have two graphs. One is the CFTR activation, and the other one is the CFTR inhibition. We can clearly see both conditional knockout and the CF group show nearly zero chloride movement in the absorptive direction, and the CFTR specific CFTR, the ionocyte specific CFTR deletion exactly phenocopies the CF group. So we conclude that pulmonary ionocyte-mediated chloride absorption solely dependent on the CFTR. Then next question is about how does ionocyte contribute to the chloride secretion? And similarly, we performed the Wilson chamber experiment. So we reversed the chloride gradient. We started experiment from symmetrical no chloride and moved to a no apical chloride and a high basolateral chloride to make a driving force towards secretion. So similarly, three groups here are control, conditional knockout, and CF. We can clearly see the control group showed a very large amount of chloride movement from the basolateral side to the apical side in an absorptive direction. CF showed an absence of chloride movement, and the ionocyte-specific CFTR deletion showed a reduced chloride secretion. Then by quantification, we can clearly see the CF group showed a zero chloride secretion, and the ionocyte-specific CFTR deletion showed a significantly reduced chloride secretion, and the reduced amount is about 70% reduction when compared to the control group. So those Woosen chamber experiments are really under short circuit current conditions. So which means the voltage is climbed to zero. So we can only make a conclusion on pulmonary ionocyte regulates both chloride absorption and secretion through CFTR under short circuit conditions. So next, we are trying to address how does the ionocyte work in an open circuit condition. So which is under more natural voltage and the physiological condition. So I think the best approach is workload swelling assay, and we did a workload swelling assay to measure the fluid secretion. So two groups here, one is the control group and the other is ionocyte-specific CFTR deletion. So I'm going to play the video here. So in presence of amenorrhea and phosphorine, those organoids in control group, they really are capable to swell because of fluid secretion. However, when we move to the ionocyte-specific CFTR deletion, we can see the workload swelling field in a CFTR condition knockout group. And then further, we check the ionocyte presence in those organoids. We found the ionocyte presence in our organoids. Then next, we move to the quantification. Here we can clearly see the CFTR specific, the ionocyte specific CFTR deletion showed a absence of organoid swelling. And more interestingly, when you compare those two groups, the conditional knockout group under DMSO, which is control, to amenorrhea and phosphorine, you can clearly see the conditional knockout showed some like shrinking phenotype. So which indicates the ionocyte, the fluid absorption there should be a ionocyte independent fluid absorption pathway in the airway epithelium. So we conclude that pulmonary ionocyte regulate fluid secretion through CFTR in an open circuit condition, so by airway organoid swelling. So the next question we're trying to address is that could there be subtypes of ionocyte responsible for regulating both absorption and secretion? So here we're going to use the cystic fibrosis ferret model we have. So we use two ionocyte markers. One is ATP61G3 and the other one is BSND. To visualize those ionocytes in the ferret tracheal epithelium. So we have two groups here. One is the wild type and the other one is the cystic fibrosis group. As we can see in the control group, there are presence of ATP61G3 positive ionocyte and the BSND positive ionocytes. But moving to the CF group, we can clearly see in the image the ATP61G3 positive ionocyte is very significantly reduced. However, the BSND positive ionocytes in the CF, it remains a very similar level to the wild type. Then by quantification, we found that the ATP6 V1G3 positive ionocytes in the CF is significantly reduced. And this really makes us think about they have different flavor of ionocyte present in the CF epithelium. And those ionocytes could be ATP61G3 negative. Then next we move to the intropulmonary airway of the CF ferrets. We found that in the intropulmonary airway, there's a huge ATP61G3 positive ionocyte hyperplasia in the CF group. So as you can see, the ionocyte clusters are are expanded when you compare to the control. Then we move into the high magnification image to check the ionocyte location specifically at CF submucosal glands. We found that those ATP61G3 ionocytes present in those submucosal gland tubules and collecting ducts. Then by quantification, we found out that in the CF group, the ATP61G3 ionocyte clusters are significantly expanded, and the ionocyte numbers are significantly expanded as well. So we conclude there's ionocyte hyperplasia in the CF intrapulmonary airway. So next we check the other ion transporters such as NKCC1. NKCC is mostly involved in salt secretion. So here we did a co-localization with NKCC1 and ATP61G3 in both wild type and the cystic fibrosis group. What we found here is that a control group showed a really good co-localization of NKCC1, ATP61G3. Then move to the CF group, and we found out there the ATP61G3 gene is kind of being turned off. They showed a lot of NKCC1 positive and ATP61G3 negative ionocytes in the trachea epithelium. Then move to a high magnification image to identify those ionocytes. So as we can see in the upper panel, there's very good NKCC1 positive and ATP61G3 negative ionocytes. And we can also see the two different flavor of ionocyte in the same image. So those are the NKCC1 positive, ATP61G3 negative, and NKCC1 positive, ATP61G3 positive ionocytes. Then next we move to the nasal epithelium. So this is a high magnification image from the olfactory epithelium and the respiratory epithelium. So we can clearly find the two flavor of ionocyte present in the epithelium. So those are the CFTR positive, ATP61G3 negative, and the CFTR positive, ATP61G3 positive ionocytes there. So then how the different flavor ionocyte contribute to absorption and secretion, so it's still being open question to be addressed in the future. So then come to my conclusion summary. Pulmonary ionocytes are enriched in CFTR gene and express the CFTR protein and the apical cap pattern. And the pulmonary ionocytes regulate both chloride absorption and secretion through CFTR under short circuit current conditions. And the pulmonary ionocyte regulate fluid absorption by using the ASL volume challenge assay and secretion by using the airway organoid volume assay. And the two subtypes of ionocyte present in the airway epithelium. So acknowledgement. So I'm very grateful to have my group members, and also special thanks to my faculty mentor, Dr. John Engelhardt and his lab. And also thanks the Cystic Fibrosis Research Center, Dr. Brian Davis, for recruiting me into UAB and other CFRC faculties. And I'm very lucky to be funded by the Cystic Fibrosis Research Institute to help me to survive the faculty funding. Thanks the Cystic Fibrosis Foundation and UAB, the new faculty startup funding, and our animal models, Ferret Research Center, resource center for providing all the CF ferrets.